Good morning, everybody. My name is Cornelia, and I'm the product manager for gene editing here at Takara Bio Europe. So thanks for joining today's webinar, in which I would like to focus on the benefits of Cas9 guide RNA RNP delivery for gene editing experiments. So I hope you can hear me well, and you will be muted during the webinar, but you can type your questions into the question box at any time, and we will have time to discuss those at the end. Okay, so let's get started. So the typical CRISPR-Cas workflow is composed of 5 major steps, and starting with the guide RNA design and efficacy screening. And this is of course a very important step because you would like to use a highly specific and also a highly efficient guide RNA. And there are many online tools nowadays to design such guide RNAs. The next step is then of course the delivery of Cas9 and the guide RNA into your cells. And here the most important points to consider are of course to achieve very efficient delivery and minimal toxicity to get efficient editing. And here the method of delivery may also depend on the cell type you are using, because not all methods are efficient in all cell types. And another important point is of course also to minimize off-target effects during delivery. And during this webinar I would like to focus on this step of the workflow. Following the gene editing, there are also several tools for a quick screening of the cell population, just to quickly check if the editing has been efficient prior to the single cell cloning step, which can be a quite tedious step and a long step in the workflow, in which you will need to maximize the survival of your single clones. And then last but not least, you can screen your single clones to identify those that carry the desired mutation, and you might also wish to screen for off-target effects to check the specificity of your gene editing event. And then you will end up and isolate and expand an edited single cell. So during this webinar I just would like to go again step by step through this workflow and show you tools that Takara Bio offers, and as I said, with a focus on the delivery part of this whole workflow. So for the guide RNA design, what I would recommend is to design 4 to 5 guides per target gene. And so as I said, there are many in silico tools that are very helpful to design your guide RNAs, but I would also strongly recommend an efficacy screening prior to the cloning and delivery of your guide RNAs to your cells. And we have a very simple test that helps you to get rid of inefficient guide RNAs and to only clone and deliver those that work. And this is a purely in vitro test that I would like to explain here. So in the first step, you can produce your own guide RNAs using in vitro transcription. And it's a very easy principle of just designing a very long oligo, which will introduce a T7 RNA polymerase promoter and your target-specific guide RNA sequence, as well as a little bit of homology to the guide RNA scaffold. In the next step, you will then just perform PCR to create a template for in vitro transcription. And this template contains, of course, the T7 RNA polymerase promoter and then the guide RNA sequence, the target-specific sequence and the scaffold sequence. And in the last step, you can then just in vitro transcribe your specific guide RNAs. And those guide RNAs can be used either for the delivery directly into cells or for the in vitro screening. And the in vitro screening is also very simple. It's a quite fast method because there are no cloning steps involved. You will amplify the target region of your gene by PCR, including, of course, the PAM sequence. And then you will just combine in a tube recombinant Cas9 that is provided in the screening kit with the guide RNA you in vitro transcribed in the first step and with your PCR product. And if this guide RNA efficiently binds to the target sequence, then Cas9 will be able to cut in vitro, and you can see this cleavage products on a gel. And this is just a very easy screen of different guide RNAs to get rid of the ones that do not cut, because guide RNAs that don't cut in vitro will not cut in your cells neither, and so it's not worth to spend time to clone and deliver those into your cells. So now, of course, you want to deliver your guides and the Cas9 to the cells. And as I said in the beginning, the choice of the delivery method might also depend on your target cells. If you have very easy to transfect cells, plasmid delivery can be a good choice. For in vivo applications, for example, I would recommend AAV, which is a non-integrating viral vector. If you are more looking on screening guide RNA libraries, lentivirus is a very good choice because you can achieve stable expression of Cas9 and you will have very few variations. You might also plan to deliver Cas9 mRNA directly together with synthetic guides or in vitro transcribed sgRNA. And last but not least, also protein delivery is possible. And today I would like to show you two examples of protein delivery or RNP delivery, so delivery of ribonucleoprotein complexes of the active Cas9 protein together with in vitro transcribed guide RNA. And it has been already shown in the past by several publications, and I just choose a few of those, that the delivery of Cas9 protein together with the guide RNA has several advantages compared to plasmid delivery, for example. And I just would like to show you some data, for example, from the paper from Kim et al. So what the authors did is that they either electroporated Cas9 protein together with a guide RNA, or they did plasmid transfection. And what you can see here is that one difference is, of course, the time frame of the presence of the protein in the target cells. So when delivering Cas9 protein, you can see that the protein will be present very fast, but it will be also degraded very quickly. And after 24 hours, most of the Cas9 protein has been degraded. Whereas when using plasmid, you can see that it takes a little time to get the protein expressed, but then it will also hang around for several days in the cell. Now, when looking at the gene editing events, which are here shown by the appearance of a lower band here on these gels, you can also see that the onset of gene editing is faster when using RNP delivery. And even more interestingly, the same authors have also shown that RNP delivery compared to plasmid delivery helps to reduce off-target effects. And the authors think that this is due to this very short burst of activity of the Cas9 protein when using RNP delivery. So what they did is they targeted 3 different genes, the EMX1 gene, the AAVS1 locus, and 2 different off-target sites for this locus here. You can see that they differ in a few bases, and also 1 off-target site for the EMX1 gene. And again, they just did a side-by-side comparison of RNP delivery and plasmid, and what you can see here is that with the plasmid there are still quite high editing events at the off-target sites, whereas the RNP delivery gives higher specificity. The others also use deep sequencing of the on-target site and the off-target sites to have a better quantification of the on-off ratio. And when you just look at these numbers here, so RNP delivery is in blue, plasmid in green, you can see that the ratio of on versus off-target is much higher when using RNP delivery compared to plasmid delivery. So here we have 14, 18, and even 114 or 279 versus 1.8, 5, or 12. And in this publication here from Liang and collaborators, they observed the same as in the paper from Kim. Again, when you are, so here this was lipid transfection, but the authors also used electroporation for other cell types, here in HEC293 T cells. And again, you can see that with DNA delivery, the onset takes longer, but then the Cas9 protein will be present for several days in the cells, whereas with protein delivery and also RNA delivery, the onset is much earlier, and the protein will be degraded quickly. And they also quantified the off-target mutations using sequencing, and here you can see the ratio is the other way around. So it's the off-on ratio, and the lowest ratio is observed when using protein delivery. So these papers just show that RNP delivery can give you really highly specific gene editing and lower off-target effects compared to plasmid delivery. And so as I said, I would like to present you two different methods for RNP delivery, and the first tool that we offer are CRISPR-Cas jasicles that can be used to deliver Cas9 ribonucleoprotein complexes. So what are jasicles? The name comes from nanovesicles and glycoprotein, because jasicles are cell-derived vesicles which are made by a producer cell line after overexpression of a specific glycoprotein. And such jasicles can be used to deliver proteins, but also other macromolecular cargos that will be carried inside the vesicle. And as the jasicles also express VSV-G on their surface, they can be delivered to a similar range of cells as would be expected for VSV-G pseudotype lentivirus. So we provide a so-called jasicle production system in which you can produce your own jasicles that will contain both active Cas9 protein and your target-specific guide RNA. So jasicle production is a bit similar to lentiviral production, and you will use a 293T derived producer cell line that you will just co-transfect with a plasmid encoding your guide RNA sequence, and with a so-called jasicle packaging mix, which is provided in the kit, and that contains a lyophilized transfection reagent, and then lyophilized vectors encoding the nanovesicle-inducing glycoprotein, vector encoding Cas9 endonuclease, and the vector encoding a membrane-bound red fluorescent protein called cherry picker. So the overexpression of the glycoprotein in the producer cell line will then induce the jasicle formation on the cell membrane. And we also use another clone-tech technology, which is called idimerase, to increase the concentration of Cas9 within the jasicles. So it's an artificial dimerization technology in which we will just tether the Cas9 to the membrane-bound cherry picker fluorescent protein using small dimerization tags and a little dimerizer molecule. And this is just to enrich the Cas9 at the cell membrane and within the jasicles, and to avoid that Cas9 will go to the nucleus via the nuclear localization signal. So next step is that the loaded jasicles will just pinch off the cells and can be collected from the cell culture supernatant, similar to lentivirus. And the jasicles can then either be used immediately or stored at -70 for over 1 year. When you apply the jasicles to the target cells, they will just fuse with the cell membrane and release the active Cas9 protein together with the guide RNA for the gene editing. So to summarize this workflow, what you will get in the kit are these mixes with the packaging, the packaging mix, the jasical packaging mixes, and you will get a plasmid to clone in your target-specific guide RNA. And you will just need to add this plasmid to the packaging mix 1, add the mix 1 to the mix 2, and then do this big transfection of the producer cells in the presence of the little dimerizer molecule. You remember this is just to enrich Cas9 within the jasicles. Then you wait 2 to 3 days and you can collect the jasicles from the supernatant, and there's a centrifugation step to concentrate them, and then they are ready for use. So now what's nice with the fluorescent protein is that it not only helps to enrich Cas9 within the jasicles, it will also indicate efficient delivery to your target cells, because when you see that your target cells get red, it's a good indication of successful delivery. However, the red fluorescence will not show you if you have really efficiently packed Cas9 into your jasicles. And for this, one easy way to do so is just to do a Western blot in which you will lyse part of your jasical production and do a Western blot with a Cas9 antibody and, for example, compare to recombinant Cas9 if you have, or which quantities you have packed into your jasicles. And here you can see that we have approximately 200 nanograms of Cas9 in 5 microliters of jasical production. You can also see that it migrates a bit higher, the Cas9 and the jasicles, and this is due to the dimerization tag compared to the recombinant Cas9. So I would like to show you some data in different cell lines about the efficiency of jasical mediated gene editing. And here we try to knock out a ZS green cassette in various cell lines and compared side by side to plasmid delivery. So on the left you have easy to transfect cell types, and on the right hard to transfect cell types. And what you can see is on the left that in this very easy to transfect cell types, sometimes plasmid delivery gives higher gene editing efficiencies, sometimes jasicles work better. In all the cases you should get lower off-target effects when using jasicles compared to plasmid delivery. And now the most interesting cell types are, of course, those that are very difficult to transfect. And here you can clearly see that we could achieve good gene editing efficiencies of 20 to 30% with the jasicles, whereas plasmid gave really very low or inexisting gene editing efficiencies. The only cell line which was really very resistant to any type of editing was the KBM7 cell line. In this example here, we knocked out an endogenous gene, so not this was not a knockout in stable clones, but the endogenous CD81 receptor. So this is a nice model because it's a surface receptor that you can easily label using a FITC conjugated antibody, for example. And then the fluorescent signal in the flow cytometer will give you an indication of the presence of the surface receptor on the cell membrane. And so this would be here, and in case of a leftwards shift of the peak, this indicates a functional knockout of the protein. And you can see here on the left with vesicles we could achieve high knockout in Jurkat cells, whereas plasmid delivery was very inefficient for the knockout. And this is partly also due to the fact that Jurkat cells are difficult to transfect. Another example of a bit more tricky cell types are human-induced pluripotent stem cells. And here again we knocked out the CD81 receptor. So you have here the positive control of the expression of the receptor and the negative control just with the autofluorescence of the cells. And we treated the iPS cells just following the standard protocol with a bit longer incubation with the vesicles, 6 or 24 hours instead of the 4 hours in the standard protocol. And then we measured the CD81 levels 7 days later by fax. What you could see is that we could achieve between 40 to 50% functional knockout, shown here by this leftward shift of the peak. To culture the human iPS cells, we used our Salatis DevCS culture system, which is a complete culture system for stem cells that also allows single cell passaging and single cell cloning. And interestingly, following the vesicle delivery, we did not see any toxicity and no effect on the pluripotency of the stem cells. So vesicles are also really suitable for difficult or sensitive cell types. So now the last point I would like to mention with regards to vesicles are that we also did some experiments with regards to off-target effects. And we compared plasmid delivery to vesicle delivery side by side for the knockout of the EMX1 gene. And you can see here the potential off-target site that exists that has 3 bases difference to the on-target site. So now when using plasmid delivery, so again here the cleavage indicates genome editing. You can see here genome editing events at the EMX1 site, but also cleavage bands at the off-target site. And now when using vesicles, we had comparable results or efficiency for the EMX1 site, but you can see that the off-target site here is very clean and comparable to the negative control. And this was also clear on the sequence the sequences of single clones. Again, here on the left we have the EMX1 site, and on the right the off-target site, and here is plasmid delivery, and on the bottom vesicle delivery. So you can see here when looking at the sequences when using plasmid delivery, there are many gene editing events in the different clones here, but also when sequencing the off-target site you can see that there are many indels. Whereas with the vesicle delivery there was specific editing at the EMX1 site, and all the clones we sequenced were wild type for the off-target sites. So this really shows that we found the same result as in the previous publications that underlined that RNP delivery can help to reduce off-target effects. So now I would like to present you the second method to deliver protein, Cas9 protein together with in vitro transcribed guide RNAs. And this is electroporation of recombinant Cas9. And I just showed here the Lonza nucleofactor or the neon from Thermo Fisher. And what we provide is highly pure recombinant sgCas9 carrying a C-terminal nuclear localization signal. So our enzyme has high concentration, 3 micrograms per microliter, and low glycerol content, 10%. So why is this important? So the high concentration is critical when using electroporation because you usually use very low quantities, low volume. And the low glycerol helps to get really high electroporation efficiencies. And compared to other recombinant Cas9 enzymes that are on the market, we are, to my knowledge, the only one to provide a 10% glycerol enzyme. The other enzymes are all provided in 50% glycerol. And in the following slides I would like to show you that electroporation can be also used to target hard-to-edit cells, including hematopoietic stem cells and also human iPS cells. And we provide protocols for the neon transfection system and the 4D nucleofactor system from Lonza in the protocol of the recombinant protein. And what we also found is that we achieved very high efficiency in combination with guide RNAs produced with the in vitro transcription kit that I explained at the beginning. Okay, so here are some data for electroporation. So we used human iPS cells. So this is the cell line 18. And we electroporated so in vitro transcribed guide RNA combined with a recombinant protein against 3 different target sites, CCR5, C4BPB, and CYP2E1. And then we screened the cells with a mutation detection kit that I will explain a bit later, 2 or 6 days later, and we calculated the gene editing efficiencies. And you can see here, so of course the efficiency depends on the target site and also on the guide RNA, of course. We could achieve between 14 and 58% of editing efficiency. And there was not so much increase on 2 or day 6 on the screening. There was not a big difference when screening the cell 6 days later instead of 2 days. We also knocked out again the CD81 target, and here we did not measure on the genome, we did not use a genome-based method to measure the knockout, but functional knockout. And again, we used the iPS cell line 18 and also the cell line 22. And what you can see here, so this is the control showing the CD81 expression at the surface, and what you can see here, we could achieve very high functional knockout, so over 85% with this electroporation experiment. And this was measured 5 days after electroporation. So functional knockout is always higher compared to this mutation detection analysis because this mutation detection assay will detect mismatches, and it always underestimates a bit the gene editing efficiency. We also compared our recombinant Cas9, here okay, you can see the result, for the CD81 knockout in stem cells with other enzymes that are on the market. And so for our conditions, we combined our enzyme with guide RNA in vitro transcribed with our kit. And for the other enzymes, we combined the recombinant protein with synthetic CRISPR and tracer RNAs as recommended by the providers. And what you can see here is that we could achieve best efficiency in the iPS cell line when using our enzyme together with the in vitro transcribed guide RNA. So much higher genome editing efficiencies. Last but not least, I also would like to show you some knock-in data, and here in CD34+ hematopoietic stem cells using electroporation. So these cells are known to be very difficult to edit and difficult also the delivery is quite difficult. So what we did here is we used a single-stranded DNA repair oligo with either HIND3 or HIND3 plus BAMH1 restriction sites. And the repair oligos had 90 base pairs homology arms. And we could achieve 2 to 10% knock-in efficiency as shown here by these restrictions, these bands that appear. So knock-in efficiency is always much lower compared to knockout efficiency, and 2 to 10% is really not bad in this cell type. What you can also see is this band here is due to the existing BAMH1 site in the CXCR4 locus. And interestingly, the CD34 plus cells maintained this phenotype. So we measured CD34 expression 5 days after the genome editing, and you can see here that still 98% showed CD34 expression, indicating the stemness of the hematopoietic stem cells. So the electroporation did not influence this pluripotency of the stem cells. Okay, just to finish with this whole workflow, we already saw some pictures of this gene editing efficiency check, and I just would like to explain how this tool works to quickly screen a cell population downstream of the gene editing. So our kit is called the mutation detection kit, and it's a mismatch detection method. And you already saw several gel pictures where always the appearance of lower bands indicates successful gene editing. And I just would like to explain how this works. So in the first step, you will amplify the target region. And since following gene editing, there are many different indels, you will get a mixed population of PCR fragments. Some will be wild type and some will carry the different mutations. So what's nice with our kit is that it contains a DNA polymerase which is called Terra that can be used for direct PCR. So you do not need to purify your genomic DNA. You can just start with a crude extract. The next step will be simply to denature and re-anneal the DNA. So all the molecules will be a bit scrambled, and you will end up with, for example, some wild-type molecules that will re-anneal with a mutated molecule, or 2 different mutations. And this will create a mismatch. And the enzyme which is provided in the kit, which is called the guided resolvase, is an enzyme that specifically cleaves mismatched DNA. And such cleavage products can then be analyzed on an agarose gel, and you can use densitometric analysis and a formula to calculate the genome editing efficiency in your cell population. So this is really a tool for a mixed population, not for single clones, and it's a very fast screen because you can complete the whole protocol within 4 to 5 hours. And if this screening is successful or shows good gene editing efficiency, you can go and go for the single cell cloning, which I said already in the beginning, which is a quite tedious step and which can take several weeks. And depending on the cells you are using, this can be very difficult because, for example, we have the experience with the pluripotent stem cells that those cells really do not like to be isolated, and pluripotent stem cells traditionally grow in colonies. But you cannot screen a colony. This is very time consuming. And the problem with those kinds of cells is that the single cells will just die or differentiate. So there's a real need for single cell culture systems of pluripotent stem cells. And we provide the Cellatis DevCS culture system, and this is really a complete culture system that allows single cell passaging, but also single cell cloning downstream of gene editing. So if you are working with iPS cells and interested in single cell cloning, there will be a webinar on the 18th of May that will really focus on this aspects. So it's just about single cell cloning downstream of gene editing of human iPS cells. So don't hesitate to drop me an email and ask for the link for registration if you are interested in this webinar. Let's get to the last step of the workflow. So once you have isolated your single clones, you can then screen them to check which ones carry the desired mutation. And here we have one tool which is called the Guided Genotype Confirmation Kit, which is a quick screening of single clones. This kit can be used to identify wild-type clones and clones carrying monoallelic versus biallelic mutations. So how does it work? You will again amplify the target region, and then you will just incubate everything in vitro with recombinant Cas9 and with a guide RNA, for example, your in vitro transcribed guide RNA. And if your clone, for example, is still wild type, so if it's not modified, the original guide RNA can bind again and there will be an in vitro cleavage. If your clone carries just one allele that is modified, the original guide RNA will be able to bind on the wild type allele but not the modified allele, and you should get one large fragment and two small fragments. And if your clone carries a biallelic mutation, those are most often the most interesting ones, you should not get any cleavage, and this can be easily analyzed on an agarose gel. And then you can decide, for example, just to sequence all biallelic clones. You can already do a quick screen on all your clones and then sequence the biallelic ones. And for example, here we also have a tool just to amplify clone and prepare these target sites for DNA sequence analysis, and this tool is called the Indel Identification Kit. Yeah, I hope that I could show you that jasicles and electroporation are two great tools to effectively deliver Cas9 guide RNA RNP complexes to a broad range of cell types, and also including difficult to transfect and non-dividing cells. So both methods can be used for knockouts, and electroporation is also suitable for homology-directed repair. And the major advantage really of the RNP delivery is that there's this short burst of activity of the Cas9 protein. So there's no coding sequence present, so no chance for genomic integration and no persistent nuclease overexpression. And this really helps to reduce off-target effects. Yeah, thanks for your attention. I will now check if there are any questions that we can discuss. Just 1 second. Okay, so the first question is, what about plant or fungal gene editing? Do we have kits or methods to do editing here? So jasicles will not be suitable for plants or fungi because they contain VSVG, and so it's a broad mammalian tropism. I recently saw a paper that's in which the authors delivered recombinant Cas9 to plant protoplasts, I think from Arabidopsis and rice. So we have not validated our recombinant Cas9 for this purpose, but I saw this publication, so I think technically this electroporation or they used, I think, PEG transfection of recombinant protein can be possible. And our in vitro screening and also the downstream mutation detection, genotype confirmation and indel identification kits are also suitable for plants or fungi because they contain the steropolymerase, and you can also do direct PCR on such samples. Okay, then there was another question, just one second. So one question is, following the vesicle delivery, how long will the Cas9 protein stay in the target cells? So we have not measured this exactly, but according to the publications, the protein is really expected to be degraded very rapidly, and most of the protein should be degraded in 24 to 48 hours. Of course, this also depends on the target cell type and the proteasomal activity. But yeah, we should expect just a very short burst of activity. Okay, there's another question. The knockout efficiency for CD81 in the iPS cells was higher with electroporation in comparison to vesicles. This result might be target dependent, or do you always have higher efficiency with electroporation? No, I don't think that you have always higher efficiency with electroporation. It may really depend also on the day of the experiment when you culture your cells. Both are highly efficient in iPS cells. I think the data I showed you for the vesicles, so this was not done side by side. You're right, we had about 40%, and for the electroporation it was over 80%. But I think to really evaluate this, you would need to do a side-by-side comparison. And of course also for different, so this was, both experiments were for CD81, but also depending on the guide RNA, you also could see that for other targets sometimes the efficiency is lower. It also depends, of course, on the guide RNA used. And here for the both results I showed you, I'm not sure if it was exactly the same guide RNA, for example. Another question regarding the guide RNA screening system. The question is, what is roughly the percentage of guide RNAs discarded after the in vitro testing? This is a bit difficult to answer, but because of course it will depend on your specific guide RNA sequence, and it's a bit difficult to predict. But from our experience, usually 1 to 2 guide RNAs out of 4 do not perform very well and can be discarded. Another question is, how can I select positive cells after the vesicle delivery? So this can be, you can enrich the cells that express the cherry picker using flow cytometry. So it's a quite easy way to enrich positive cells. Okay, so I do not see any further questions. Also, if you have questions later on, don't hesitate to drop me an email. And the PDF version of the slides and the recording will also be available shortly. So thank you very much for listening, and I wish you a nice day. Bye-bye.